Okay, everybody, today we are talking about how is it that psychedelics work? A lot of my information is from MAPS's most recent lecture in their webinar series entitled Psychedelics in the Brain, but I also did a little bit of research into basic neurobiology for you. So let's get into it. What and how do psychedelics work? Psilocybin and LSD, they have very similar effects, but they achieve those effects by very different means. And this is, for all my non-nurses, referring to the mechanism of action of each drug. The mechanism of action is basically the biochemical ways that a drug or a molecule causes its pharmacological effects in the body. So the how. Let's talk about psilocybin and LSD and how they're a little bit different. Psilocybin is just simply a serotonin agonist. LSD is a serotonin and dopamine agonist. But what exactly does it mean to be an agonist? An agonist is essentially a chemical that bonds to a receptor and then causes some sort of action. You contrast that with an antagonist, it basically does the opposite. An antagonist blocks a receptor and therefore inhibits or completely stops an action. So here's my little picture. So agonist bonds to the receptor here and it causes an action. Antagonist, it blocks the receptor and causes no action. Very simple, right? Now, let's talk about something a little more complicated. We're going to talk about neurons, synapses, neurotransmitters, receptors, the whole thing. So, what exactly is a synapse? A synapse is the space between two different neurons, and it's how they communicate with each other. It's also called the junction. So, the two different neurons, the presynaptic neuron is the one that happens before the junction, and the postsynaptic neuron happens after it. And the way that they communicate with each other is through electrical impulses or through chemicals that we know as neurotransmitters. Here's another little picture. Both gray areas are neurons. So this is the presynaptic neuron we just talked about and the postsynaptic neuron. All these little dots, those are neurotransmitters. And essentially what you see is those neurotransmitters are being released by the presynaptic neuron, and then they're binding to receptors on the postsynaptic neuron. And then they cause some chemical reactions. Actually, not that difficult, right? So, what is going on with our mushrooms, Molly, and acid? So, serotonin and dopamine are the main neurotransmitters that we are concerned with when it comes to these drugs. But more importantly, specifically for psychedelics, it's not the neurotransmitter itself, it's the receptors that it bonds to. So let's contrast MDMA with our psychedelics, mushrooms, and acid. So, you know how you always hear about people doing Molly and feeling depleted? You don't really hear that with the psychedelics like mushrooms and acid, and the reason has to do with the different mechanisms of action of the drugs. So MDMA, how does it work? It actually operates just by simply increasing the amount of serotonin in the body. You contrast this with mushrooms and acid. What they're doing is they're actually interacting with the receptor for serotonin and to a lesser extent dopamine. So how this works. Psilocybin and LSD are chemically similar enough to serotonin and dopamine that they are able to bond to serotonin and dopamine's receptors. And then they're causing effects that are much different than the serotonin or the dopamine would have caused. So contrast MDMA, it's completely just increasing the amount of serotonin. LSD and mushrooms, they're kind of mimicking serotonin and dopamine, but causing very different effects in the body. So this is why with psychedelics, you get visuals because they're causing different effects than the serotonin would have. And with MDMA, you're just getting a flood of the happy feelings and the euphoria because you're just net increasing the amount of serotonin. I should clarify though, small caveat. It's not that MDMA is just magically producing all this increased amount of serotonin. It's not a magic thing. What it's actually doing is causing the reuptake of serotonin to be blocked. So if you're binding to a receptor that's then blocking the transporter that's involved with the reuptake by the neuron of serotonin, because basically neurotransmitters, they'll be reuptaked by the neuron to create the full cycle. We're blocking the reuptake of that serotonin, so then it's increasing the amount of serotonin available in that synapse. So that's kind of the nitty-gritty of how the net increase occurs. Now let's talk about exactly how psychedelics work in the brain. So there's a few theories out there, there's a few hypotheses, but the most prominent one that's kind of been tested the most is called the thalamic filtering model. But first, let's talk about just how the brain works. So when you look at the brain has been mapped out so well that we can look at a specific region and know how that region affects the body and our cognition and everything, right? How do we see the world? So there's two different main types of areas in the brain that we're going to talk about here. There's the visual areas, and then there's the associative areas. So the visual areas, the sensory areas, what's going on here is that the sensory areas are taking in not just visual. I should preface this. It's sensory areas and associative areas. And those sensory areas, they're taking in visuals, they're taking in auditory, they're taking in touch, anything that has to do with the senses, they're taking in all that information. And what the associative areas do is they make sense of that information. So they're integrating all of that great sensory information and they're making sense of it in the brain. So again, to recap, sensory areas, associative areas. Sensory areas are taking in all the sensory information. Associative areas, they're just making sense of it. They're integrating it in your brain. So how do we take in sensory information? Basically, it comes through the brainstem and then it goes to what's called the thalamus. And the thalamus has this really unique function. It's often referred to as the filter, the gatekeeper, or what's the other one? The relay center. Sorry, excuse me, looking at my notes. So basically, information comes in through the brainstem, goes to the thalamus, and the thalamus decides what is important to pass on to the other parts of the brain. It connects that part of the brain, that like very primitive part of the brain, with the higher aspects of the brain, the cortex. And it's saying what information needs to be passed on, what is important. So can you imagine what's maybe happening with psychedelics? With that thalamus, that function, that normal function of telling the brain what's important and what's not important, it's basically out of funk. So the thalamus and its filtering capacity, its relay center, its ability to decide what's important and what's not important, it's compromised when we take psychedelics. And what is the net effect? You have an overload of sensory info. Your brain is just rich with what's going on in the world. It is all coming in. It's coming in a little confused. You might see sound. You might taste color. It's all kind of jumbled up. So how did researchers test this hypothesis? I am only going to speak about one study, but it's really interesting. And if you'd like to know the link for the study, just let me know and I'll send it to you. So how did researchers test the thalamic filtering model? They administered LSD to participants and then basically put them into an MRI, a functional MRI, so that you could see which parts of the brain were being activated. And this might sound a little scary, but actually no one reported any sort of bad experience. Everybody was enjoying their trip in the MRI. So what did it show? In support of the hypothesis, the researchers found that while on an acid trip, the thalamus is more strongly connected to those sensory areas, and it's actually less connected to the associative areas of the brain. Meaning we have this flood of sensory info that we kind of guessed we would with this hypothesis. Interestingly, you have this flood of info, but you don't have the ability with those associative areas because those are turned down when you're on a psychedelic dose on a trip of acid. You can't integrate. So you're just flooded and you have no means of integrating what it all means. So this is very important because it augments our sense of reality, right? We aren't capable of integrating information in the same way when we're on psychedelics. So that's the therapeutic potential that underscores psychedelics. If you augment reality, you can change the way that people think about themselves, about the world, about everything that's going on in their life. You might be able to rewire some patterns of thinking that aren't really serving that person. Now, I should say that this study that I just spoke about, it was the psychedelic doses of acid, meaning a high enough dose that you're tripping out in that MRI, right? But we're often hearing nowadays about microdoses and the importance of microdosing. So what those same researchers did is they administered LSD, but at a microdose and did the same functional MRIs. And what they found was very interesting. The thalamus maintained its normal function as the relay center, as the gatekeeper. It worked fine. There was no change in the amount of connection between the thalamus and those sensory areas. So you didn't have that flood of visual, auditory, tactile information. However, the region of the brain that was definitely stimulated more was the amygdala. And the amygdala is essentially our emotional center of our brain. It's how we regulate our emotions. So there was basically a greater connectivity between the amygdala and the frontal regions of the brain. And let me say, the frontal regions of the brain are actually associative areas. The ones that were downplayed in the psychedelic doses were actually more connected with the microdoses. So why is this important? Those associative areas help us integrate information in our brain. And during those microdosing trips, during those microdosing experiences, what a lot of the participants explained that they felt was just a little bit of a sense of a positive mood. So if our amygdala, which is the region of emotional integration in our brain, is being activated to a higher extent while taking a microdose, and we also have this positive mood that's being reported by participants, if we can find a bridge between the positive mood, the amygdala, which is our emotional center, and then these associative regions that are helping us integrate into our brain, that is a very profound potential for microdosing of LSD. So, taken together, these two studies show us a few things. One, they show us that psychedelics are indeed dose dependent, which obviously if you're tripping balls during a psychedelic dose and you're just very minorly even at all affected by a microdose and just feeling a positive increase in your mood, clearly that makes a lot of sense, right? Like most medications and most drugs, psychedelics are dose dependent, meaning in higher doses you feel one sort of set of symptoms and in lower doses you feel another set of symptoms, or effects, I should say. The other aspect that we need to underscore here is that these two studies show that psychedelics can work in both the sensory parts of our brain and the associative parts of our brain. So essentially, we just have barely scratched the surface of how we can utilize psychedelics. And what we really need is more studies to bulk up our evidence. We need to test more hypotheses than just the thalamic filtering model. And we also need to be able to reproduce results. I'm talking about just two isolated studies. For science to really have an impact, we need to have results that are reproducible and valid under any circumstance. So that ends my little lecture on how psychedelics work. I hope you learned something. And, just let me know if you have any other questions.